Sorry, sir.
Okay, very good morning, my dear friends. This is Dr. PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So, in the regard of CPGET-2023 PG online free coaching for Botany and Plant Sciences for all competitive exams also, JL, DL, Gurukula DL, JL, PGT also, my dear friends. So, this is the wonderful opportunity. So, my dear young friends, those who are completed your degree examination or degree final year examinations, those who are applied for CPGET 2023, and also the aspirants of the JL, DL of other competitive examinations, this coaching is very useful for you. So, eminent resource persons are going to deliver their lectures. In this regard, today's our eminent resource person, very huge experience personality, eminent botanist, Dr.A.Kavitha Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College, Autonomous, Hyderabad. Madam, please welcome and share your screen and start your session. Madam. Please welcome.
Okay, sir. Thank you very much, sir. Good morning to all the participants over here. Now, from today onwards, we are going to start with the type studies of the Pteridophyta. So, one of the type study of the Pteridophyta, that is Lycopodium, will start from today. I'm just sharing my screen. So I'm just, yeah. So we are starting with today with the classification of the Lycopodium and all the morphological, anatomical, and the reproductive characters of the first type study of Pteridophyta, that is the Lycopodium. Now, when we look into the systematic position of this Lycopodium, it belongs to the division Lycophyta. So before going to that, I want to just mention a few points regarding the classification of pteridophyte. The pteridophytes, according to Smith classification, they have been divided into 4 divisions. Now, what are those 4 divisions? One is called as Coelophyta. Second one is called as Lycophyta. Third one is called as Sphenophyta. And the 4th division is called as Pterophyta, or we can also call it as Filicophyta. Okay, so the complete pteridophytes, they are divided into 4 divisions. So Lycopodium comes under the 2nd division of pteridophyte according to Smith classification, that is Lycophyta. Now this division Lycophyta, it is again classified into 2 classes. One is called as Ligulopsida and another one is called as Lignopsida. So these are the 2 classes. So Lycopodium among that belongs to the class Ligulopsida. And Ligulopsida, this is a class which is divided into further only one order. So that order, what we call it as Lycopodiales. And this order consists of only one family, that is Lycopodiaceae. And under this family Lycopodiaceae, we are moving with the study of Lycopodium. So I'll repeat this classification again. Lycopodium belongs to the division Lycophyta, the class Ligulopsida, order Lycopodiales, family Lycopodiaceae, and the genus Lycopodium. So there are around, along with Lycopodium, this family also includes another genus which is called as Phyloglossum also. So that is not there in this now at UG level. Now, this genus Lycopodium, it is divided, it consists of around 180 species. And these are considered to be as the living Lycopodiales, and they are representing during the period of Carboniferous period of a geological time scale. And many of these types, so many of the types, then they are going, we call them as Lepidodendron, which were the large trees. Okay, so the Lycopodium, that is the fossil pteridophytes we represent is as the Lepidodendron, so which were the large trees. Now, the living one, the modern representatives of this Lycopodium, they are very small and herbaceous sporophytes are present. The leaves are very small and simple, and each leaf they possess an unbranched midrib, and the leaves they are going to have, that does not have any ligules. There are no leaf gaps in the stele of the stem, and that's why the leaves are very closely, compactly arranged on the central stem. And the sporophylls, they may or may not be restricted to the terminal part. Now, these sporophylls also, they are simple, having the vegetative leaves, and they possess only the homosporous sporangia, and the gametophyte, they are wholly or partly subterranean, that is, they may be that may be underground or coming out of the ground system. And the antheridia of this gametophyte, they are remaining embedded in the tissue of the prothallus, and it develops anthrozoids. Now look into this here. This is the Lycopodium plant. You can look into this. The tips of these branches, we call them as the sporophylls. Okay, that we are calling, so this is the strobulus. The group of these sporophylls forming a cone-like structure, we call them as the strobulus. Now, so the species of this Lycopodium, they are worldwide in distribution. They are mainly found in tropical and subtropical forest. And in India, we found to be they grow in the hills of the eastern Himalayas. So there are many commonly occurring Lycopodium species, which are called as club mosses or ground pines, or we call them as trailing evergreens, etc. And many of these species, they occurs in the hanging epiphytes. They appears like hanging epiphytes. So some of the species, if we talk about of Lycopodium, the examples like Lycopodium clavatum, Lycopodium cernum, Lycopodium setaceum, Lycopodium phlegmarium, serratum, philanthum, etc. But among all these, the common species are always available. That is Lycopodium clavatum and also Lycopodium cernum. Now, as I told you that pteridophyte, the dominant phase or the adult plant body, it is represented or it is designated as the sporophyte. Now, so when we talk about the habit, that is how the plant body appears. The species of Lycopodium, they are very small. They are herbaceous, or we can call them as the shrubby sporophytes. I repeat, these are small, herbaceous, or shrubby sporophytes. The stem in almost all the species, it is delicate and weak. So that's why they appear to be, you can look into this here. The stem, it is not erect. It is weak and delicate. So these species appear to be growing as epiphytic condition with erect or pendant sporophyte, while some other species, they are terrestrial and having a trailing habitat. That is, they appear to be like vines. As the stem is very weak, they appear to be growing on the support. So we call them as the vine. The stem and its branches, they are densely covered with the small leaves. And these leaves, they are very compactly arranged. So when we talk about, they are sessile, very compactly arranged on the stem part. Now, when we move on to the stem, the structure of the stem. So the stem is referred to the subgenus Eurostachya, which is possessing branched or unbranched stems that are erect or pendant. Okay, so when we observe in case of Lycopodium clavatum, the stem is dichotomously branched, and those branches, they are unequal in size. There are certain branches which are smaller in size and they are weak, and they bear certain strobili at their tip region. So this type of branching in Lycopodium clavatum, we call that as pseudomonopodial branching. So the species here, you can look into this, Lycopodium selago, lucidulum, phlegmaria, and others, whereas it is dichotomously branched, which is observed in, and that too, the dichotomously branched is unequal, showing a pseudomonopodial branching in case of Lycopodium clavatum. So when we go for the anatomy part of this stem, right, when we talk about the anatomy of the Lycopodium stem here, right. So this is a TS or the anatomy of the Lycopodium. We are taking the example of Lycopodium clavitum here. Now, so when we observe here, the transverse section going to consist of a thin layer of parenchymatous cells with the cutinized outer walls and stomata. So this is the outer epidermal region you can see here. This is the outer epidermis. This is going to be covered with the cuticle, and it is going to consist of stomata here and there. And this epidermis, it is going to followed by a thick layer. You can look into this here. This is a thick layer of cells, which is called as hypodermis. Or we can also represent it as outer cortex. This is the outermost part of the anatomy, that is the epidermal part. It consists of here and there the stomata and highly cuticalized. It consists of the cuticle and cells are parenchymatous here. Below this, you can see a very thick layer of cells, what we call it as a hypodermis, or we can also call it as the outer cortex. Next, this hypodermis, it is going to be followed by many layers of thin-walled parenchymatous cells, which is going to be called as the middle cortex. This is the outer cortex or hypodermis, we can call it as. And this region is represented by a parenchymatous thin-walled cells, which is a little bit majority of that is covered. And this can be called it as the middle cortex region. And when we move on to the deeper into the anatomy of the stem here, there is a thick walled. So you can see here, there is a single layer of a thick walled inner cortex, we call it as, or this is called as the endodermis. So here in this endodermis, it is without any intercellular spaces and it is continuous. Now, below this endodermal region, you can see here, this is the complete sclerenchymatous cortex. This is a peculiar characteristic feature of the Lycopodium. Below the endodermis, you can see a huge sclerenchymatous cells region, which is called to be as the sclerenchymatous cortex also. So which is going to form the underground tissue of the Lycopodium clavitum. Now, next, if we move on to the central part of or the stellar or the vascular region of this transverse section of the stem here. Now, the pericycle here, you can look into this. Below this sclerenchymatous cortex region, there is a pericycle which is consisting of 1 to 2 layers. You can see this is one layer and the second layer. It is represented by only 1 to 2 layered of parenchymatous cells. Now, below this pericycle here, we can see the vascular region, which is a compact zone consisting of primary xylem and the primary phloem. So I'll show you here. So this is the vascular strand. Okay, represented. This is the phloem and this is the xylem strands. Now, so when we talk about the Lycopodium species, each Lycopodium species shows different types of steels. Like, for example, you can look into this here. The first one, this one. This is the stellar region of Lycopodium serratum. Now, in this Lycopodium serratum, you can see here that this type of stele is called as the actinostele condition. Now, why we are calling it as the actinostele condition? Because here the xylem strands are in the form of a star-shaped structure. This one and also this one. Okay, they are in the form of a star-shaped structure, where this is the protoxylem. The protoxylem is going to be situated at the tip region of these star-shaped projections. I repeat this once again. The example or the species called as Lycopodium serratum or Lycopodium anotinum. In this two species, the stele condition, the stellar region is called as actinostele, because the xylem strands here, they are in the form of a star-shaped structure with the protoxylem at the projecting ends of this star shape. Okay. This can also be observed in Phlegmalia and Selago also. Now, next we are moving on to the another type of the stele, the species called as Lycopodium cernum. Okay, so this is the Lycopodium cernum. Okay, so in the Lycopodium cernum, in the Lycopodium volubuli or in the Lycopodium clavitum. Okay, so this is the Lycopodium volubuli, the D1. This is the species of Lycopodium volubuli or Lycopodium clavitum. In these two species, you can see here, these are the xylem strands. These xylem strands are in the form of the plates. They are like in the form of plate-like structures, and the protoxylem here is in the tip regions of these radiating plates. Okay, so this type of stele is called as plectostelic condition. This type of stele, we call it as plectostelic condition. Examples are Lycopodium. These examples, they are often asked in the exams. Actinostelic condition is observed in which species of Lycopodium? It is Lycopodium serratum or Lycopodium anotium. Or plectostelic condition is observed in which species? Lycopodium clavitum and Lycopodium volubula. So when we move on to this type of stele in cernum, in the Lycopodium cernum, here you can observe this stele is called as a mixed protostelic condition, where the xylem as well as the phloem, they are uniformly distributed throughout the stellar region. Okay, so here you can see this is the xylem which is occurring in the form of irregular tracheids. That is the xylem we can see here. It is appeared to be scattered in the complete mass of the phloem tissue. And that means to say xylem as well as phloem, they are uniformly distributed. Xylem is scattered. It appears to be the xylem is scattered in the phloem tissue. So this type of mixed protostelic condition is observed in the species called as cernum. And generally what we can say that the vascular strand or the vascular tissue of the Lycopodium is considered to be as the protostelic condition, which is consisting of primary xylem and the primary phloem. Right. So here you can see this is a Lycopodium clavitum. I told you, this is an example of a Lycopodium clavitum where you can see the xylem strands. These are the xylem strands which are in the form of plates, plate-like structures distributed in the phloem tissue, distributed in the phloem tissue. So this is about the transverse section of the or anatomy of the Lycopodium stem. Now we are moving on to the leaves. So you can see here, this is the Lycopodium stem, and you can observe, or these are the branches. You can see here this complete stem and the rise and these branches, they are surrounded by the leaves, which are very small. So now when we talk about the description of the leaves are numerous in number. They are many in number. They are very small. They are simple and they are very sessile. That does not have any petiole. You can look into this structure here. That doesn't have any petiole. They are sessile. They are very small and the leaves are simple leaves. And one more important point here to be remembered in Lycopodium means The leaves are closely and compactly arranged, covering the entire stem and the entire branches. Okay, so that to be remembered. So very closely and compactly arranged of the leaves. Now, so these leaves, they are generally about 2 to 10 millimeters long, and they are closely arranged in spirals. Spirals in the sense, you can look into this here. Spiral arrangement, that is, come entirely to the complete stem or the branches is going to be covered with the leaves. Spirally arranged in the examples like clavitum and the anotinum, while in other cases they are arranged in whorls like in circles. Example is cernum. In some species, the leaves are found to be arranged in opposite pairs. This is the opposite pair. So this is a spiral arrangement. This is a whorls. Okay. Right. So in opposite pairs, the example for that is Lycopodium alpinum. So in others, they are irregularly arranged, and usually these leaves are lanceolate. So when you observe the shape of the leaf, they are pointed at the tip. You can look into this here. The tip of the leaves are pointed and they are very long. So that shape of the leaves, we call them as lanceolate leaves. Now, when we go to the anatomy of the leaf here, so when we talk about the anatomy of the leaf represent the transverse section of this leaf represent a triangular in shape. Okay, it appears more or less triangular in shape with an single layered epidermal layer with closely arranged parenchymatous cells, and it is going to be covered by a thick layer of cuticle. You can see here outer cuticle, and these are the parenchymatous cells. Okay, there in Lycopodium, though it is a stem or a leaf, it is highly cuticalized. And we can see the presence of openings in this epidermal layer. We call them as the stomata. Now, these stomata, they are distributed on both the surfaces of the leaf. And more or less here, this leaf is an isophilous leaves. In the examples like volubuli, generally they are isophilous, but in the species like campanulum or volubulum, they are anisophilous. Right. So internal to this epidermal layer, this is the internal part which we call it as the mesophyll tissue, which is undifferentiated into palisade and spongy. We can't see any differentiation of long palisade tissue and his parenchymatous spongy tissue. No, it is not like that. The complete mesophyll tissue is consisting of loosely arranged parenchymatous cells, which all these are the loosely arranged parenchymatous cells, which consist of the stomata in them for performing the photosynthesis. Now, when we talk about the central vascular part of this leaf, in the vascular region here, it is going to be consist of the xylem and the phloem, and here it is considered to be as a mesar condition. That is, phloem in the center surrounded by the xylem. So that condition is called as the mesar condition. So this is about the transverse section of the leaf. Okay, so you can see this is the branch, and this is this is the stem, and this is the branch, which are going to consisting of the leaves, which are very closely arranged, which are either spirally or whorally or irregularly arranged, covering the entire stem and the branches. Now, so next we are moving on to the root system. So the roots, they arise from the outside that arise outside the stele, and they do not penetrate in the cortical region of the stem. So these roots, they turn downward and penetrate the middle cortex, making the canal sweat, and we call them as the cortical roots. So you can see the transverse section of the root here, outer epidermal layer possessing the root hairs. Below to this is a thick walled hypodermis, we call it as the outer cortex. Below to that is a parenchymatous inner cortex region, and this is the central cylinder, vascular cylinder. And one more point here to be remembered is the vascular cylinder here. in the Lycopodium root, it can be identified with the help of the xylem, which is in the form of a horseshoe. The shape of the xylem is in the form of a horseshoe shape, or it is in the form of an inverted C. Okay, with the protoxylem projecting at the corners of this C. So that is a significant characteristic feature here of the Lycopodium root, and the central part is the phloem. So with this, when we take a transverse section here, when the vascular cylinder is appearing like this, it can be identified as the Lycopodium root. So this is the most important characteristic feature or a distinguishing feature to be remembered of Lycopodium root. Now, next we are moving on to the reproduction point of view. So even in pteridophytes, we can see the vegetative reproduction or vegetative propagation of the sporophyte can be seen. So that it can be happen with the help of a juvenile stage of the sporophyte, that can also be called as the protonema, or the vegetative propagation of the gametophyte, that is any small tissue of the gametophyte, if it is detached, that can be developed into a new adult thallus. Or the vegetative propagation can also be carried out either through gemmi or through bulbils or through tubers, which are developed at the apices of the roots. Next, we are moving on to the sporangium, that is the sexual reproduction, what we can talk it about. So the sexual reproduction in Lycopodium, it takes place with the help of the spores, and these spores, they are developing in the specialized structures, what we call it as the sporangium. And these sporangium, they are developing on the leaves, on the special leaf-like structures, what we call it as sporophylls. I repeat this. Asexual reproduction in Lycopodium, it is going to be carried out with the help of the spores. And these spores, they are developed in the specialized structures. We call them as sporangium. And these sporangium, they are developed on the leaf. Okay, they are developed on the fertile leaves, we call them as, which are representing as the sporophylls. Those leaves which are developing sporangia, those leaves are called as sporophylls. Now, so this is all about how the sporangium is developed. So the inner daughter cells are formed by this periclinal division. They give rise to the basal portion of the sporangium. The outer daughter cells, they contribute to the sporangium tissue. And the outer cells divide periclinally and forming the jacket and the inner cells. Now within the sporangium, the archisporial cells, which divide and periclinally and they form a massive sporogenous tissue. Now this tissue, they act as the spore mother cells. And these spore mother cells, they become rounded and are being separated from one another. And now each one will undergo reduction division and form a tetrad of spores. Now, how are these spores? So these are the spore structure. These are the spores. Okay. Now, so they are bring the tetrad of the spores. Now, so the sporangium, it is going to dehisce. So the mature sporangium, it is about 2 mm across and kidney shape. So this is the sporangium. It is a kidney shaped structure. It is 2 mm across. On maturity, the sporangium narrows. It will transverse the strip of the cells. The stomium is getting opened and the apical portion of it will dehisce, and the mature sporangium, it will get ruptured by the transverse slit along this stomium. This is called as the line. Okay, this is called as the stomium line. So through that line, the sporangium get ruptures and all the spores, whatever they are present inside the sporangium, they are being liberated into the surrounding environment. So this is how the sporangium is developed in the tissue, in the sporophytic tissue. So one of the cell of the leaf, we call it as sporangial initial. This sporangial initial will develop and forms the sporangium. This is the spores. All the spore mother cells. This is sporogenous tissue. They transforms into spore mother cells. Each spore mother cell will undergo reduction division and forms a tetrad of the spores. Now these spores, how are they structured? How are they present? These spores, they are very smaller spores. They are yellow in color, around 0.03 to 0.5 mm in diameter. And all the spores which are formed inside the sporangium, they are uniform in size and shape. There is no variation. Hence, these spores are considered to be as homosporous condition. I repeat, Lycopodium develop homosporous spores. That means to say, these spores are alike. They are same, uniform in size and shape. Now, these spores, upon liberation from the sporangium. So, this is the sporophyll you can see. This is a sporophyll or a strobili. All these sporophylls, they are aggregated together and forming the strobili. So this, each one is a sporophyll. You can see the sporangium here. Now from this sporangium, the spores are liberated out. So when the spores are dehisced and when they are liberated from the each sporangium, they will settle at the bottom on the ground and they will start germinating and develops into the next generation of the life cycle, which is called as gametophyte. Okay, so this is the spore structure. Okay, so this is spore, it will undergo germination like this and developing into a gametophyte. So this is the gametophyte of the Lycopodium, which is called as prothallus. That means to say a single spore will develop into this gametophytic structure, what we call it as prothallus. Now, from this formation of this prothallus, the sexual reproduction of the Lycopodium will start, or the next generation of the Lycopodium will start, which is called as gametophyte. Now, why we are calling this prothallus as a gametophyte? Because you can observe here the apices, the terminal part of this prothallus are developing the sexual organs. So you can look into this. These two are the male sexual organs called as antheridia, and these are the archegonia, the female sexual organs. So both male and the female sexual organs, they are developing within the same prothallus. Hence, Lycopodium is considered to be as monoecious condition. It is represented as the monoecious species also. Okay. So you can look into this here. Antheridium. These antheridium, they are globose structure having a primary wall, that is a surrounding wall, and we call it as a jacket layer, and they will enclose the sporospermatogenous tissue, which will develop into the male gametes, what we call them as the anthrozoids. Whereas when we come to the archegonium, so one of the superficial cell of this prothallus, by periclinal division, it will transform into the archegonium. So the mature archegonium here, it is going to consist of a globose venter and the long elongated neck region. So this neck region is going to consist of neck canal cells. So the number of these neck canal cells will vary from one species to the another species. And whereas this venter is going to consist of one venter canal cell and the egg cell. Now this egg cell acts as a female gamete. From this antheridia, the anthrozoids, they will come out and they will reach the neck. The anthrozoids will pass through this neck region and enter into the venter and get fuses with the female gamete. So this is how the fertilization has occurred. Fuses with the egg cell and get transformed into the zygote. Now this zygote, upon division, upon development, it transforms into an embryo. This is an embryo which is developed on the gametophytic tissue. Now, this embryo, so this embryo here, the development of the embryo here is considered to be as the endoscopic development, because the embryo is developed within the gametophytic tissue. Now, this embryo, upon development, upon many divisions, it gets transformed into the new sporophyte. Okay, so this is the life cycle of Lycopodium. You can look into this. Right. So this is the adult plant, the sporophyte. So these are the strobili. We call it as the strobili, where you can see all these are the sporophylls. So this is a sporophyll structure which is enclosing the sporangium. Okay, so this is the sporophyll structure, sporangium. Now, what does this sporangium develop? These sporangium will produce spores. Now, what is these spores are happening? These spores are developing and germinating and producing the gametophytic tissue, what we call it as the prothallus. So the terminal part of this prothallus, you can see they form the antheridium here. They develop the antheridium, and here they also develop the archegonium. These are the male gametes called as anthrozoids. These anthrozoids, they are moving and entering into the archegonium and fusing with the egg. And after that fertilization, this is the zygote. Now this zygote, upon development, it transforms into an embryo, and this embryo will undergo differentiation and going to form the sporophytic tissue or the adult sporophyte or a new sporophyte. So this is the life cycle. So this is a representation, graphical representation of the Lycopodium life cycle. Strobulus, sporophyll, sporangium, spore mother cells which undergo meiosis, spores, prothallus, antheridium, anthrozoid, archegonium, egg, zygote, fusion, development, embryo, and then the Lycopodium. So here you can see the sporophytic generation followed by gametophytic generation. So this type of life cycle is called as diplohaplontic life cycle, because the dominant phase is the diploid phase. The diploid life cycle is alternating with the haplontic life cycle, what we call it as a gametophyte. So diplohaplontic life cycle, and the alternation of generation here is sporophyte is alternating with the gametophyte. So this is about the type study. I'm just stop sharing of Lycopodium.
Okay, madam, thank you.
Yes, sir.
Okay, thank you very much, madam. Tomorrow, same time, we will going to meet.
Okay, sir. Thank you.
Right, madam. Thank you.
Right.
Navya ji, thank you. Huh?